afternoon, everyone, and a very warm welcome to the University of the West Indies. My name is Judith Gobin, and my task is very simple. I'm here as Head of Department of Life Sciences at the University, but I am actually representing my Dean of the Faculty of Science and Technology, Dr. Brian Coburn, who unfortunately is not able to attend. He sends his very warm wishes and has asked me to bring you these few words. Deputy Principal of the St. Augustine campus of the University, Professor Indar Ranarayan, invited speakers, members of staff, students of the University, ladies and gentlemen, again, good evening. The Faculty of Science and Technology offers robust programs in computing and information technology, chemistry, physics, mathematics and statistics, and the life sciences. Each of these disciplines can be subcategorized, sometimes several times, to narrower and even narrower pursuits. Arguably, given that life is endlessly fascinating, perhaps life sciences encompasses the most diverse collection of scientific disciplines. Just last week, the faculty hosted the 1985 Nobel laureate in physics, and he alluded to the fact that biology is in fact very confounding. I'm sure if we were to poll around the room for everyone's personal favorite confounding question, there is one that would show up on perhaps everyone's cards. Who am I? I imagine everyone here must have asked themselves that question at one time or another. And apart from the information carried in our names, what was there to know? Today, the news headlines are often dominated by stories of populism and the rise of ultra-conservative interests. Much of that is in turn said to be driven by waves of refugees and the so-called economic migrants. Where did all these people come from? And more importantly, where are they heading? Will they put down lasting roots? Will they be assimilated so that in a couple of generations it will be unclear where they actually came from? Who am I? Inherited mostly intact across time, what clues lie in my DNA and in your DNA? Well, the state of the art does not currently allow us to resurrect velociraptors, and T-Rexes, but there's a great deal that can be gleaned from our DNA using current technology. Some of the theory behind that available technology currently forms part of our own thought program within the FST. This evening, you will be guided through what is possible and how it is done by our most interesting speakers and panelists. Who am I? The question is so fundamental that I am sure many of those questions will be generated and perhaps even answered. I bid you again a very warm welcome to the University, and I'm going to introduce you now to the chair of the session, Dr. Alana Roach. She's an assistant professor at St. George's University in Grenada. She's also a geneticist, and I'm very proud to say a UWI alumna. Dr. Roach.
Good afternoon all, and welcome, a very warm welcome to our discussion tonight on genomic ancestry mapping in Trinidad and Tobago. Who am I? That is the question of the night that we're trying to answer. We're also trying to answer a couple more questions. If I know who I am, if I understand my genomics, what does that say about me in terms of my health? Tonight we have two wonderful and esteemed colleagues who are here to really touch on those two topics. The first of our colleagues, Dr. Regini Haramse, will be speaking about mapping variation and ancestry of the human genome. Dr. Harak Singh is a lecturer in the Faculty of Science and Technologies. She is a graduate of Stanford University. My computer is giving me a little technical glitch. 1 second. There's so much to say about it, I have to make sure that I have it up here, right? So she also leads a research group in human genomics and personalized medicine. Her research focuses on developing and implementing cutting-edge technologies for the study of the human genome and for discovering novel genetic contributors to various human diseases. In particular, Dr. Harak Singh is focused on applying these technologies for improving diagnostics and therapeutics in Trinidad and Tobago and, of course, across the entire Caribbean. Dr. Harak Singh's expertise is widely sought after regionally and internationally. This lady is currently the Director of Panomics at Yale Transdisciplinary Collaborative Center for Health Disparities Research. She is also Vice President of patient research at the Rare Genomics Institute, which is an international non-profit organization that uses genome sequencing technology to gain biological insights into rare disorders and improve patient outcomes. As I mentioned before, she did her postdoctoral training at Stanford in genomics and personalized medicine, and before that, she did her PhD in human genetics at Yale University. Even more impressive, she has received 2 bachelor's degrees from MIT in mathematics and in biology. Her professional aspirations are to become a leading researcher, educator, and advocate for the advancement of the Caribbean through science and technologies. Dr. Harak Singh, please join us.
Good evening. I'm really glad to see so many people here today. I really had no idea what to expect when we invited the public to come and have this discussion with us, so I'm really pleased to see all of you. I study the human genome, as Alana just mentioned, and I think it's one of the most fascinating entities around. Our genomes hold secrets to our history and our health. And today I want to spend some time talking to you about what can be known from our genomes, what currently is known about our genomes, what this has to do with us as a population, especially in the Caribbean and Trinidad and Tobago, and more importantly, what it has to do with our health. So, the most fascinating thing to me about the genome is the fact that it's comprised of this molecule called DNA. And DNA, we've known since the 50s, was a hereditary, the molecule that transmits anything hereditary. But it wasn't until very recently, until about 2001, that we were actually able to read the information that's in our DNA. So our DNA is a molecule that looks like this. It's a helical molecule, and it's basically a linear molecule. And it's made up of a series of letters. You can think of it as letters. And if we were to read those letters, it would look like this, just, a string of letters. But the interesting thing about this is that these letters in this DNA actually houses all of the instructions, all of the information that make us who we are. And not only that, it also tells us how to make who we are. So our DNA, and many of you may know this already, is housed in our cells in 23 pairs of chromosomes. And these chromosomes, 23 pairs of chromosomes that are linear molecules, can be unraveled and we can actually read what they say. And this is what houses all of the information to make us who we are. So, in my opinion, one of the most interesting things about living in the time that we live in is the fact that we are able now to read the information that exists in our human genome. And as we said before, we've asked for since humanity began, we've been asking the question, who am I? And for the first time in our history, and in the time when we live, we get to experience this revolution in science, where we can actually read this information and try to figure out what it means. Now, reading the information is one thing, and we do have the technology to do that. We even have technology now to edit the information in our genomes, although, while technically possible, it's still in its infancy. But the real key issue here is not what we read and understand, but really, what does it all mean? And what has lagged behind since the first draft of the Human Genome was produced in 2001, which was a huge triumph for humanity, that for the first time ever in our history, we were actually able to read the full sequence of the human genome. We had to ask the question, well, now that we can read this, what does it actually mean? So some surprising things came out of the Human Genome Project. Well, I'm not going to tell you all of it, but a couple things. The first is, you've heard of genes, right? So, we know our genes are in our DNA, and DNA comprises our genome. It turns out that only about 2% of our DNA is actually comprised of genes. The rest of the DNA, which we've learned over the last 17 years since the Human Genome Project has been produced, actually does a lot of work to regulate how those genes are expressed. So the rest of the sequence, the other 98% of the sequences in our genome, actually play a key function in when the genes are turned on and off, and how often, and so on. And as you know, different cells in our bodies have different functions, they look different, and different cells in our body carry the same DNA. So in some of our cells, certain genes are turned on, and in other of our cells, different genes are turned on. And this is what causes some of these differences between different cell types. So the cells that exist on my skin look different from the cells that are in my liver, and part of that is because of different genes that are turned on and off. So even though the information that's in all of our cells is the same, this 98% of the genome that regulates how those genes are expressed is really what underlies some of these differences that we see in terms of our traits. So after we studied the human genome, and this was a project that took $3 billion US, 10 years, hundreds of scientists. We now had just one sequence, right? And we really wanted to ask the question, well, how different are different people's genomes? And what do those differences mean? We know if we just look around the room that everybody looks quite different. On the one hand, we're all human and we all look like humans. On the other hand, each of us is unique in our own ways. We're unique in our physical traits and how we look. We're unique in how we interact with the environment. We're unique in our health and our histories. And we're unique, of course, in our ancestry. So subsequent to the Human Genome Project, there was a big drive in the field of genomics to try to understand how genomes vary from different people around the world. And so there were several projects to this, and one of the most important ones is called the 1000 Genomes Project. And this project actually studied thousands of genomes around the world and asked the question, how different are these genomes from each other? So it was some of my PhD work went into this, but this was a huge collaboration with about 500 scientists. So on the papers produced by this project, and listed there as one of 500. But nonetheless, it was a really exciting project to be a part of, because for the first time I'm describing how different people are based on their genomes. So these are some of the populations that were studied in that project across the world. And I just wanted to point out the fact that there's even population of African Caribbean ancestry in Barbados as part of this project. So we now have this unprecedented amount of fundamental information about ourselves, about our history and our health. And this was previously hidden. It's something that we were not able to previously read before. And so what does all of this mean? The truth is, while the technology exists for us to read the information, we still have a lot of work to do to interpret what that means. And this interpretation has a lot to do with differences that we see in our various traits, including traits related to our health. So, how can two genomes differ? Well, let me just explain very briefly. So I told you before we have our DNA housed in these chromosomes. And these chromosomes are actually packed really tightly. So this DNA is a linear molecule that's packed up really tightly into a chromosome. And if we were to unravel that, and sort of like pulling a thread off of a spool of thread, you would see that if it unravels, it comes down to this level where we can read the single letters. And so we can take two genomes and line them up and see that they're almost the same except for, in this case, a single nucleotide variant. That's what that's called. So a single variation in one of the letters. And there are about 1 million of those types of variations in each one of our genomes. There are also other types of variations where we have small insertions or deletions of a few nucleotides or a few letters into one sequence compared to the other. And again, there are hundreds of thousands of those in our genomes. And then there's larger structural variation that exists between our genomes. So if you took my genome, which I told you is a linear molecule, and your genome, another linear molecule, line them up next to each other, we can compare, are the letters the same as we walk along this string of letters? And in some cases, if we have a reference sequence and a different sequence, a sequence that we're testing, we might find that large chunks of a particular string in our DNA is actually missing, and that's a deletion. We might also find that some chunk of my reference DNA is actually duplicated. We might find that those duplications occur many different times. They might occur right next to each other, or they might occur very far away. And they might be novel sequences in your DNA that are not shown in mine, are not present in mine. We also know that in some people we have sequences that are actually flipped in orientation. And then we also have situations where on some of our chromosomes, one piece of one chromosome breaks off and attaches to another chromosome. So these are some of the variety of different types of ways that our genomes can differ from each other. Nonetheless, we're all here and we're all healthy and alive. So these differences underlie a lot about differences that we express amongst ourselves. And in order for us to interpret the genome and try to figure out what is the relationship between our genes or our genetic information and the traits that we express or show, we have to understand what this variation means, right? So I want to introduce you to two concepts in genetics. So it'll be a little bit of a lecture type thing. And we're going to use these two concepts to basically understand the relationship between why it is that we see the issue of ancestry is important, especially in our population. So this concept is called identity by descent, and I'm going to explain it to you in terms that you understand, and that is, we all know that we are most similar to the people who we are most closely related to, right? So we're most similar to our siblings or our parents, our grandparents, and so on. And the less related you become to someone, the less similar you are, right? This is something that we know and understand. Now, this can be demonstrated by this concept of identity by descent in genetic terms. So first of all, let me tell you what this diagram is. So a square means a man, and a circle means a woman, and a line between them and lines coming down from that means those are their children. Alright? So here we have a man and a woman, they have two children, and this person married this person and had a child, and this person married this person and had a child. This is essentially what we're looking at here, right? So what I'm showing you in these squares, these are chromosomes, and just one pair of chromosomes. I told you we have 23 pairs, but let's just look at one pair. So in this person, they have one chromosome that has a particular sequence and another chromosome that has another sequence, and the mother has two different chromosomes as well. And what happens in this process called meiosis, which is how we generate gametes, which is how we do reproduction, you have some rearrangement of the sequences in these chromosomes. And so this child now inherits a chromosome from the mother and a chromosome from the father, but the chromosomes that they inherit from either parent is not identical to either one of the chromosomes that come from the parents. It's a mix, right? Essentially, it's a mix of the parents' two chromosomes. So if we use that principle, we can see that as we follow these chromosomes through the different generations, what we have here are actually two cousins, because their parents are siblings, right? We have two cousins, and they share segments of their genome, but they're sharing less of their genome than these two siblings. And as we go down many more generations, people who are related are going to share less and less of their genome. So the idea is that people whose ancestors have come from the same part of the world, or who have come from geographically isolated populations, are going to share more of their DNA with each other than they are versus if you compare them to someone from a different part of the world. So that's one concept I want to explain to you. And the other concept I want to explain to you is something called the founder effect. So if we have a bunch of people in some population, and some person here has a mutation, and this person has this mutation, it's pretty low frequency in this population, right? This is 1 out of maybe 50 people. But if we take some subset of this population without knowing who has or doesn't have this mutation, and we move them somewhere, and then they start to multiply, what happens is because we took a small number of people from this original population, and the frequency of this mutation increased in that was high in this subset of people who moved here, we end up with a larger frequency of that mutation in new population. Now this is essentially what may have been happening as we have taken people from Africa and bring them to Trinidad, people from India or Syria or wherever, all the places where we came from. So this may be one thing that might be at play in our population. We don't know. We haven't done any studies to test this. But this is a very well-established genetic principle that very well might be having some effects in our population. So I think our slide pointer no longer works. Okay, so what does this have to do with ancestry? Well, both of those principles can be used to basically explain the fact that our ancestry, or our genetic ancestry, I should say, is reflected in our genomes. And what I'm showing here, this was one study that was done in 2008, where a number of people from different parts of Europe, different countries of Europe, were sampled, and their genomes were sampled. And then what we did is we tried to cluster those people according to how similar their genomes were. And what it turned out to be is that all of the people from the Iberian Peninsula clustered together, all of the people from Scandinavia clustered together, all of the people from Germany clustered together, and so on, and all the people from Italy kind of clustered together. And it shows that the genetic information actually was able to recapitulate the geography that was already known historically about these people. So the information in our genomes actually is reflective of our ancestry. And the methods to do this involve various statistical methods that are statistical and computer science methods that are employed to read this data and to analyze it and to begin to cluster it into these clusters of people who are similar to each other. So what does this have to do with us and ours? Well, let me see. I'm going to go to the next video.
Is it a battery?
So, in terms of human disease, our ancestry actually has a lot to say about what's going on. So there's kind of two paradigms for how we can try to understand the genetics of human disease. One is that we have everybody who has the same disease has the same mutation. And this is the case in some diseases, like sickle cell anemia, for example. Everybody who has this disease has the exact same genetic mutation, which we understand quite well and how that causes that disease. And there's another situation where everyone has the same disease sort of on a physiological level. Let's call it breast cancer, meaning that tumors are growing in the breast. But on the genetic level, this cancer is actually caused by very different causes, very different molecular causes in each individual with this disease. So even though they seem to have the same disease, they actually have a very different, or they have unique conditions if you describe it on the molecular level or on the genetic level. And what happens in reality is that these two paradigms are actually both displayed in our population. So in some cases, for some diseases, we have some subset of people that have the same mutation that's causing their disease, and then we have other individuals who have a totally different cause for this disease. And now you might be wondering, how can it be that two people have the same disease and have a totally different cause on a genetic level? So I'll show you a very simple way of that. So the answer is that genes don't work in isolation. Genes work in biological pathways. Genes work in biological pathways, and they work together. So here's one example. This gene makes a protein. All genes make proteins, and the proteins are the things in the cell that actually do the work. So the protein might be a receptor on the cell surface that interacts with a hormone or a drug that causes some sort of change to that cell. The protein can also be structural. Protein can also be an enzyme that carries out some reaction in a cell. So protein A activates protein B. Protein B inhibits protein C. And protein C activates protein D. And protein D causes some trait, right? So this is a pathway. And this is what's going on. Now, you can imagine what's going to happen if C is not there, or if there's a mutation that renders C ineffective. There's no C, so that means nothing's activating D, and so that trait isn't being produced. Yeah or no? All right. Is there another way to not produce this trait? Okay, well, what if there was no A? If there's no A, then there would be no B, and nothing would be inhibiting C. So C would activate B and we would produce trait. So there are other ways. So we can see from this example that if we had a mutation in one of these genes, or in another one of these genes, we can eventually produce the same outcome, even if it's not the same mutation. That's the point of this. What actually happens in reality is that we don't have just 4 genes. We have, in the human genome, we have 25,000 genes. And these 25,000 genes work in very complex networks. And it looks something like this for you to try to map it. It's called a radiculogram. Because it looks ridiculous, right? It's just all of these interactions trying to be mapped onto each other. But because of this complexity, what comes out of that, the main point of this is that we can have 2 people who have the same disease, but have different causes on the genetic level, different mutations that are causing the same disease. So we know that our health is a product of our genes and our environment, right? So what's hard-coded in our DNA, as well as how we interact with the environment. And what we know about our situation in Trinidad and Tobago is that our disease burden is extremely high. So about this is some major causes of death in Trinidad and Tobago, according to the World Health Organization. And about a third of the deaths in Trinidad are due to cardiovascular disease. Another 15% or so is due to cancers, and another 14% is due to diabetes, complications of these diseases. So almost 2/3 of our mortality is actually due to these non-communicable complex human diseases. So what does that have to do with our ancestry and our genetics? Well, we know that these disparities exist in our propensities to develop certain diseases. And I'll show you a little bit now about some of the data that has come about so far about what we know about this phenomenon and why that means that we need to study our population. So here's some recent data about cancer incidence and mortality in Trinidad and Tobago. This is basically showing that cancer incidence is actually much higher in people of African descent than in people of Indian or mixed ancestry. And the mortality is also much higher. So why is that important? Well, cancer is caused by a number of different genes. I'm going to use the example of breast cancer. Many of you may have heard of the BRCA gene, which Angelina Jolie famously had a mutation in that gene and opted for a double mastectomy before she developed any symptoms of breast cancer. But this is just a picture of the BRCA gene, one of the BRCA genes. And this is showing that there are something like 15,000 different mutations in this gene. And these mutations actually differ in their frequencies across people of different ethnicities. And the thing is, this BRCA mutation is one of those things that is so well known that there are now genetic tests and panels that people can pay a fee for and have themselves tested to see if you have mutations in this gene. Now, this kind of testing has been very successful in certain populations, especially Caucasian populations, in the Ashkenazi Jew population, and so on. And there's currently the most, extensive genetic test for breast cancer is now offered by a company called Fzero Genomics. And people in Trinidad have some access to this test. There's actually a study that's ongoing at NRC where participants can get tested for free. And there's also, you can also pay for the service, I believe, at one of the pathology labs in Trinidad. But the thing is, a lot of people that have breast cancer and clearly have hereditary breast cancer, meaning that it runs in their family and so likely is caused by a mutation. A lot of these people, it turns out, when they get tested for this gene for BRCA or any of the other known genes that cause breast cancer, it turns out they don't have mutations in those genes at all. So why is it that they have no mutations in those genes, yet they still have some form of hereditary breast cancer? And our hypothesis is that it's because they have mutations in other genes, and we are trying to look for what those other genes might be. So here's one example. So this is one family pedigree that came to us that when patients underwent testing on this most extensive breast cancer genetic panel, they came back with absolutely no mutations in any of those genes. So it means that there may be mutations in other genes that were not tested, and we are trying to find what those are right now. But the thing is, why do they not have mutations in those genes? And why is it that panel can be sold and marketed as, the most extensive breast cancer panel, yet it's not working for these people? It's because it was developed based on people of a certain ancestry, and those people do not necessarily have the same mutations that are causing this disease in people of different ancestry. So essentially, these tests that we see marketed, and you will, as time goes on, these tests are going to become more and more popular. We have to kind of, I think, have a discussion and talk to you and educate our public about the fact that some of these tests are not actually going to be relevant to you, and they're not going to be useful. Now we don't know which ones are going to be useful and which ones are not going to be useful, and that's part of the complexity of the issue. The only way we can know that is if we study our own population. And so what you're going to find is that in the next few years, lots of people are going to come at you and ask you to participate in this genetic study or that genetic study. I'm going to ask you that because we have several studies going on. You may have seen some of my PhD students out there in the health centers and so on, trying to get you to participate in some of our genetic studies. And companies are going to come at you asking you to pay a fee to learn some information about yourself. And you as an individual are going to have to decide how relevant is that to me, and how much money am I willing to put out for this, and what am I going to do with this information, and is it actually going to mean anything to me? So this is what's going on with breast cancer. A similar thing is going on with diabetes. This is another project we're working on, where we see families that clearly have diabetes running in their family like this. And we think that this might actually be due to some founder mutations. There's anecdotal evidence that in families of East Indian origin in this country, the rates of diabetes seem to be higher. Now, we can't actually find comprehensive data even epidemiological data that can confirm this, but we've seen, this is based on a number of different sources of anecdotal evidence. Nonetheless, the idea is, we think there may be higher frequencies of these mutations that may be causing diabetes in this population because of the fact that people have been brought from elsewhere and have sort of bred amongst themselves and increased the frequency of these mutations in our small population compared to the ancestral populations from where they came. And the last story I want to tell you about is about a heart disease story. This is in collaboration with Naveen's teacher and the Faculty of Medical Sciences. So, how many of you have heard of the drug Plavix? Some of you may be on this drug, right? Plavix is actually the first-line drug that is given when someone comes in with a heart condition that requires blood thinning. And there are a few pieces of evidence here to show that we can actually, if we are able to actually implement genetic testing in our health system for certain mutations, we might actually be able to improve the situation. And let me explain why. So, the generic name for Plavix is clopidogrel, and this is a drug that essentially thins your blood. And there are certain mutations in the genes that metabolize this drug. So when you ingest the drug, it gets metabolized, and that's due to certain enzymes, right? So the genes that encode these enzymes have certain mutations that sometimes make these enzymes not work. And so if you have this mutation, that drug is not going to be effective on you, because you will essentially be resistant to that drug. Now, it's been shown in other populations around the world that, in fact, it's even in the algorithm that doctors use to prescribe this drug. So in the US, for example, if you're considering antiplatelet therapy, you will first be genotyped for this gene, meaning you'll try to figure out if you have this mutation or not. And if you have certain versions of that gene, you'll be given the standard dose of the drug. But if you have other versions of the gene that may render you resistant to that drug, you're actually immediately started on a different drug. Now, as you can imagine, that's not the situation that's going on here. And Naveen has in his clinic at Mount Hope has recently completed a study that shows that about 60% of about 50% of his patients are actually resistant to this drug. And how he discovers that is by, checking. He prescribes them the drug, and they go off and live their life, and then they come back to their next doctor's appointment, however many months away that is, and they get all their testing done again, and we notice that they're not improving, right? But there are actually biochemical tests that we can do. So he's done some biochemical tests that actually show that these patients' platelets do not show the behavior that they're supposed to show when this drug is administered. So we can actually test biochemically whether or not these people are resistant to this drug. And it turned out about 60% of patients of Indian origin in Trinidad are actually resistant to this drug. And there's some other data from Munger or another FMS study that shows that the frequency of these specific mutations that are relevant to that resistance are actually higher in Indo-Trinidadians than in Afro-Trinidadians. So what we're trying to do now is make this link to see if this increased frequency of this mutation is actually what's driving this resistance to this drug in this population. So what I told you were just 3 stories about how health disparities that we observe in terms of, disparities in terms of disease prevalence based on genetic ancestry might actually play a role in how we deal with diagnosing or treating certain diseases in this country. And we're talking about 3 of the biggest killers amongst us. The other issue I wanted to just touch on briefly is that sometimes, or all of these studies, rely on something called self-reported ancestry. So when I go in to participate in a study, I'm going to say, I'm Indian. I've actually tested my genome and it says it's 99.9% Indian. But, you know, some of us may not actually even know what, you know, we have in this country a number of different interesting mixes. And some of us may not even know what our full ancestry might be. And why is that important? It's important because if you want to say that a certain mutation is relevant to you, you have to take into account the ancestral genetic background, because in some populations that mutation may confer some traits or some outcome, and in other populations it may have no effect. So, in summary, what I told you about today were 3 things. We have all these advances in sequencing technology and DNA sequencing technology that are allowing us to read all of this information that is on genomes. And it's now apparent that any 2 humans are quite different, right? Our genomes vary substantially more than what was previously thought. And these differences are what underlies these differences in our traits. What's also important is that because of these differences, we're starting to understand disease on a molecular level that is driving us towards a future where a one-size-fits-all model of medicine is no longer going to apply. We're going to have to treat people based specifically on what's going on in their own biochemistry and in their own genetics. And what we've noticed as well, based on all of this data that's been accumulated across the world, as more and more genomes get sequenced, is that ethnic variations represent some of the most divergent examples of human variation. And so ancestry is extremely important when we are trying to understand genetics. So this means that the underlying genetic contributors that result in certain diseases might be similarly diverse in studies in understudied populations, because it's not recognized that the real way we're going to interpret the genome and make sense of what it means, the human genome, is to carry out studies that involve a diverse set of participants. And when we say diverse, we mean ancestrally diverse. And that's the only way that we're going to be able to unravel all of these secrets that our genome holds. So, with that, you are going to be asked in the future to participate in studies and to pay for services, and you might very well want to. But I urge you to think about, as you're being offered all of these services and these chances to participate in studies like this, you're going to have to weigh certain issues about what you want to know about yourself versus what is the utility of you participating in the study for the greater good. you're going to have to weigh issues of your own privacy and your own data being exposed versus issues about it being out there and useful to the rest of the world. So these are some things we're going to discuss in the panel, and I think I'll end there and let you speak to our next speaker.
Dr. Haraxing, thank you so much for simplifying the science of genetics and helping us understand the interplay between genetic variation and disease. We are also joined tonight by a second speaker, Dr. Dexter Hadley. Dr. Dexter Hadley works in Silicon Valley, USA. He is an assistant professor in pediatrics, pathology, and laboratory medicine at the University of California, San Francisco. His expertise is in translating big data into precision medicine and digital health. That means, basically, that when Dr. Haraxing's lab collects all of this data and all of these samples, he translates a lot of that information into modalities for treatments and therapeutics for individuals and subgroups. Dr. Hadley's background and training is in genomics and computational biology. He received his bachelor's in natural science from the New College of Florida. He then completed a master's in systems engineering, followed by a PhD in genomics and computational biology at the University of Pennsylvania. Dr. Hadley wears several hats. He is also an MD, and he later cemented his expertise in this field by completing a residency at Stanford University. His lab is heavily funded through the National Institutes of Health in the United States, where he does a lot of his work. I invite Dr. Hadley tonight to join us to help us understand how we can translate some of this big data that we're collecting on genomics and genetics into precision medicine that might be important to us to help improve our public health in Trinidad and Tobago.
Thank you, Dr. Roche. Thank you, Dr. Roxanne, for a great introduction. My name is Dexter Hadley, and as Dr. Roche said, I translate big data into precision medicine. So, can anybody tell me the difference between data and information? What is data versus what is information? If I told you number 10. If I told you number, I'm sorry?
Get info from you.
Correct. If I told you number 10, it could mean 10 years, 10 months, 10 minutes. If I told you my daughter is 10 years old, and happy Girl Child Day to everybody here, by the way, International Girl Child Day. But anyway, so my job is to translate data with numbers into information. And I'm going to tell you a story today about big data. But before that, I have to, this is just what you have to do, I have to disclose all my conflicts of interest, of which I have many. So, as Virginia said, I've been funded by the NIH, which is this big one there, and they have more money probably than all the others combined. But I've been involved in these 4 companies up on top here, that are funded and work on. Alright, so we're going to talk about precision medicine. So the first time I heard about precision medicine was in 2015, when Barack Obama got up in the State of the Union and said, this is where America is going. And I was like, what? We used to call it personalized medicine before he decided to change the name and tell anybody. But precision medicine, or personalized medicine, brings us closer to curing diseases like Dr. Haraxing spoke about, diabetes, complex diseases, diabetes, mental diseases, heart disease, cancer, big one is cancer. And the very thing about it is in a tropical context. So a febrile kid shows up to the casualty ward, fever, chills, joint pain. How do you know that kid has dengue fever that he can or she can die from, or the common cold or a flu? where you don't, it's really difficult to make that clinical diagnosis. But with precision medicine, there are biomarkers and there are molecules that we can measure, RNA, DNA, whatever it may be, to help doctors make that diagnosis. So this is a big data talk. How big is big data? We all know kilo, mega, and giga. You think giga is big data? How about terabyte? Terabyte, it's a lot of data. Maybe. So I work at petabyte scale. So petabyte is 10 to the 3, 6, 9, 12, 10 to the 15. That's 15 zeros. It's petabyte scale. Right? Twitter, Facebook, they operate on exabyte scale. So the amount of messages that we send a day, probably more than exabyte scale. They probably operate on zettabyte scale. There's a huge amount of information that we generate today. And just for completion, 10 to the 24 is 24 zeros. 24 significant digits is the octabyte scale. I think the weather, the amount of variables we collect to predict the weather operates at this scale. Massive scales. So, I have a type of petabyte scale if I'm lucky, exabyte scale. So it's not that big. It's not that big, right? What date am I talking about? Well, Dr. Braxton stole my picture, but it's DNA. From Wikipedia. Right, so DNA. DNA is 4 bases or letters as difficult. But DNA is essentially chemistry, right? So DNA is a sugar phosphate backbone with 4 potential bases. And it's a specific coding. Adenine always binds to thymine, and guanine always binds to cytosine. And that simple code, 4 bases, 2 rules to pair, really encodes all that we know as life today. From the time of the dinosaurs and before that to now. That's amazing, right? So if it's 4 letters and 2 binding rules, that becomes a computer algorithm problem. Computers are really good at solving letters and numbers. And as Dr. Roxy mentioned, this DNA, these 4 bases that are bound by certain rules, are arranged in tightly wound into 23 chromosomes. As you know, you get 1 chromosome from your mom, 1 chromosome from your dad. So that pairs, 23 pairs of chromosomes. And this pretty much encodes who you are biologically. Right? So we have DNA. What makes us different? So 2 humans on average are 99.9% odd identical. Right? So out of the 3 billion, so that's why we have the primary prefixes, out of the 23 gigabases, out of the 3 gigabases in the human body, maybe a couple 100,000 are variable among us. So there's less than 1% variability among humans. Human chimp is something like 99% identity by state, like Dr. Raxon spoke about. And you go down the evolutionary tree, human and mouse, something like 40 million years divergence, 60% similarity between person's genome and a mouse genome. So that's why we work on mice and pig and dog and various model animals to test drugs, because a lot of our DNA is the same. So, where does this variation come from? So, mutation, obviously. Mutation introduces variation into the genome, but that's not where most of the variation comes from. It comes from regular old meiosis and crossing of a homologous recombination. Because when these, when homologous chromosomes come together, one from one from and they sort their DNA as is depicted on this picture, entire lengths of DNA get inherited and changed in configuration of the sequence of each respective chromosome. Right, so homologous recombination, remember that, is responsible for the bulk of the variation in the genome. What makes us us. And as Dr. Raxon alluded to, we have technology today to read it. Sequencing is the hot topic today. Everybody wants a sequence, but a whole genome is on the order of maybe 1,000 years ago, I don't know, $2,000 or $3,000 US today. Yes, it's going down in price. But remember what we just said, right? So homologous recombination means DNA is inherited in stretches. So if I know the first letter of the stretch of DNA, I know the last letter of the stretch of DNA. I don't need to know what's in between because it's pretty constant, right? So genotyping gives us a different view of DNA than sequencing. Sequencing gives us a complete view of DNA. Genotyping gives us snapshots. So out of the 3 billion pairs of bases that we have, a couple 100,000 can be genotyped with this type of technology. We don't need to get into the details, although I probably will. So what do? You have, so now we have big numbers, we know how to count, right? So mega, giga, we know how to count, big data. We have data to count. What do you do with it? Well, you put it in a bank, put it in a biobank. This picture, I think, is from the UK Biobank. So the UK Biobank is responsible for collecting, genotyping, processing, and accounting for millions of UK citizen samples. And with this information, the National Health Systems in England or in UK decides who gets what medicine and who doesn't. Who gets clopidogrel and who doesn't. Because they have a massive information evidence base to optimize a limited resource, which is the cost of healthcare, which is not cheap, right? So this is where the big data gets stored, and this is where somebody like me gets involved. So a biobank is a type of biorepository. So it's not only the ones and zeros, not only data, but the samples that generate that data, right? So this is a $100,000, if not $1 million facility filled with robots and all kinds of double, triple guarded security doors and whatnot to make sure this data is available and understood. And that's wet data, so real biological spit, blood, urine, feces, and dry data, or ones and zeros. So with that introduction, I'm going to tell you a story about big data that I was involved in at the Children's Hospital in Philadelphia. So this is the pediatric hospital of the University of Pennsylvania. It was the first children's hospital in America, and it's still among the most prestigious and top children's hospitals in America, or pediatric hospitals in America. And bar none, CHOP has the biggest pediatric biobank of samples. When I did this maybe 10 years ago, they had, maybe 100,000 samples. Today they have over 300 samples. 300,000 samples of data from kids that go to the hospital that have blood drawn and genotypes extracted. Right? That's a lot of data. That's quite a bit significant. CHOP is a multi-million-dollar, tens of million-dollar infrastructure. They have all the robots, they have all the biobanks, they have everything in high throughput and on a large scale. And I want you to pay attention to this number. It says, so basically, every participant in this genetic study is asked if they would like to be recontacted if we find something that might be of interest to you. And 85%, almost all the participants say this, right? So just keep that in mind. So recontact is important in the biobank because it allows you to take what you learned from the biobank and go back and test it on the people you're doing it from. So as Dr. Augustine pointed out, there's single nucleotide variation, right? So single base pairs can get changed. But what I'm going to tell you about today is copy number variation. So these are the duplications and deletions in DNA, in germline DNA that were born with from our parents. You might have heard of trisomy 21. Right? So trisomy 21 is an entire chromosome 21 duplicated. So now you have 3 chromosomes and massive effects on the individual that has large-scale deletions, large-scale duplications of chromosomes. I'm not talking about entire chromosomes being duplicated, I'm talking about kilobases or so stretches of chromosomes that get duplicated. And the reason they get duplicated is the machinery of DNA replication. It's a little bit noisy, right? So if there are long repeats when the homologous chromosomes align for recombination, there may be slippage in the replication machinery, which either extends or deletes part of the chromosome, depending on which homologous chromosome we're talking about. So suffice it to say, there's micro deletions and micro duplications that exist in the human gene. And we actually did not know this before. We thought it was just at the level of chromosomes. We had no idea before the human genome was sequenced that there's so much variation in deletions and duplications. So the way this works, this is a computer chip. This is a laboratory on a chip. Right? So, extracted nucleic acid is literally put onto this chip. It's bathed in nucleic acid. It goes into a machine that looks like this. And what this machine does, and what this chip has, right, is specific primers. So remember I told you DNA is a code. There's a way these base pairs code A, T, C, and G. So you can make a complementary primer for particular targets that you're interested in, line them up in the hundreds of thousands. And then the trick is, this machine shines light on these primers. If they're complementary, meaning the target that you're looking for binds to your primer, it's going to reflect light at a different intensity. So if it's not complementary, if the primer remains unbounded, right? So this is a physics reaction, essentially, where you have a detector collecting photons, and depending on the intensity of those photons, you can estimate what the DNA is, or what the variation in the DNA is. Remember, this technology wasn't really made to develop, wasn't developed to call duplications and deletions, but it was made to call A, T, C, and G. So again, this is about 10 years old now, but I was very fortunate when I was in medical school, at the end of medical school at Buchanan and Shaw, to work with some of the cutting edge algorithms at the time to take this data that's supposed to be just A, T, C, and G. If you can get raw intensities, right? So you can measure the intensity of light reflecting and being collected by the detector, the photons. So imagine if you have increased intensity being reflected back at you, is that a deletion or a duplication? You have more light being reflected versus no light at all. So if you have more light being reflected, you have more DNA. More DNA means it's more duplication, and that's what you're seeing here. So here we have all the intensities lined up across this chromosome, which is chromosome 1, I think. Not that important, but you see the intensity here is elevated relative to the intensity here, right? So that's a micro duplication. And if you have a decrease in intensity, you have a corresponding micro deletion, right? So we train machine learning algorithms to read these raw intensities and make these biologically relevant calls 10 years ago. Why? Because think about it, you have 3 billion base pairs in the genome. Depending on who you ask, maybe 5% of them are important. That's still millions of base pairs that are potentially important. Let's say you found one that has changed out of your population or your experiment. One has changed. It's very difficult to figure out what gene is causing that change because there's 3 billion possibilities. I mean, there's a large number of possibilities. On the other hand, there's only about 20,000 genes in the human genome. So if you can predict a whole exon, the first to last intron of a gene is X size, you can imagine it's probably a little bit easier out of 20,000 targets to predict which one is functional relative to millions or tens of millions of base pair targets, right? So we want to work in gene space. A gene is the functional unit of inheritance in genetics, right? You inherit genes from your parents that, yes, there is DNA, but the point is looking for deletions and duplicates of genes is a much easier task to explain than looking for base pair deletions or base pair changes, right? So, I've been in school for a long time. I'm 40 years old and I'm still in school. And when you've been in school this length of time, your value is really measured by your peers and what they think of your work that you do. Nobody cares what I think, it's what they think. And we were able to publish this work in a very highly regarded Nature Genetic journal. And the work is essentially comparing 13,000 kids that were collected at CHOP, 3,500 had ADHD, 9,000 did not have ADHD. There had hundreds of thousands of variables, SNP markers in all these kids. And comparing these two populations, we get this. So don't get scared. What this shows is the top genes with either deletions or duplications in that contrast between cases and controls. Remember I told you there were 13,000 kids, right? 4 of them have this gene, 3 of them have that gene. There are about 20 kids in total out of 13,000 have these mutations, these deletions or duplications in genes. It's not a lot. But what is interesting about it is they're all in the same family of genes. So genes over evolution, they diverge, they diverge within species, or so-called paralogs, or they diverge across species, so-called orthologs. And again, there's slippage of the mechanism. You can imagine over a lot of time, genes get completely duplicated, and you have whole families of genes that now work together. And given there's about 20,000 genes in the genome, how unlikely do you think it is that 1, 2, 3, 4 are 4 top genes? It doesn't matter that they're very small numbers. But if you look at the significance, the p-value, they're not found in controls, right? They're found in cases, they're partly found in controls, they're not found in controls. So that's a very significant finding. And the fact that it's in one family of genes, so let's call it a super gene family for didactic purposes, is very significant. Because now, instead of looking at 20,000 genes, we have one gene family to go look at and to think, how can we fix this defect? Again, it's really difficult to make a case that for 20 kids out of 13,000 we will build drugs, billion-dollar enterprises, right? It's hard to make that case. But as Dr. Haraxing described earlier, genes don't operate in unison, right? So genes, RNA goes to DNA, DNA goes to RNA goes to protein, the ethos of genetics, but it doesn't end there. Because proteins interact with each other to do things. One protein can't do anything. So if we take this information from protein-protein interaction maps of the human genome, and we input the information we learned from our patients, cases versus controls, we read on this, or deleted genes. Every object on the screen is a gene in a protein-protein interaction network. We know GRM is important, so 20 kids is not enough. If you look at the first and second interacting neighbors of those 4 or 5 GRM genes that we find, there's now 240 genes that are involved somehow in ADHD. And if we just call them red if there's a deletion, green if there's a duplication, you can call them 2 ways, right? So you can have more deletions in controls, or you can have more deletions in cases. So in this picture, if there's more deletions in cases, meaning this mutation is enriched in cases, it gets colored dark. There's a dark red, there's a light red. And if there's more duplications in cases versus duplications in controls, they also get colored dark. There's a dark green, there is no light green. Which means, well, if you just look at the picture, it's very dark. Okay?
I'm sorry to interrupt you, but no event in Trinidad and Tobago will be completed without this kind of announcement. PDN 3046. Sneak out quietly, we don't want to see what you're blocking someone, alright? Sorry about that.
Thank you very much. So the picture is dark, and you can put a p-value on it. How we do it is not that important. You can calculate it here. And now we have 240 genes and hundreds of kids in this network that is impacted by the GRM, glutamate metabotropic receptor. The metabotropic glutamate receptor, GRM family of genes. It's expressed in the brain. It's a very good candidate if you're looking for drugs for ADHD. So this, I joined when we started with ADHD. I joined a couple of years in. I started to focus on autism, and I was able to publish the same network, metabotropic glutamate receptors, not only in ADHD, but in a very different disease, autism. So are people familiar with autism and how it's diagnosed? So you have ADHD, you are hyperactive, you are distractible, which is fine. You don't have, to have autism, you have to not have everything else. You have to not have schizophrenia, not have epilepsy, not have ADHD, and so on and so forth. If they can't find something to diagnose you with, you have autism, right? So by definition, it's a much more clinically diverse and difficult diagnosis. And we show this molecularly. So if we look at exactly the same analysis, in fact, instead of 30,000 kids, we have almost 20,000 kids now. 6,500 with autism and 12,500 without. For bigger numbers, when we look at the top genes in autism, none of them are GRM. If you look at GRM, the significance of these GRMs, they're 10% likely due to chance, not very convincing, compared to 1 in, 1,000, 10 to the minus 4 is a small number, right? So, whoa, what happened to GRM? Well, nothing. At the network level, it's still significant, right? So it's not as dark as ADHD, but the network for autism is significant for 240 genes that are connected to GRM. So it suggests that if we find a drug to treat this defective metabolic, if you find a drug to treat the defective metabolic glutaminergic receptors, it may work across the entire neuropsychiatric spectrum. It's so said, so done. So again, smoking gun, top 4 or 5 genes in the genome-wide association studies of copy number variants, shows up a gene family. A quick search in Ingenuity shows you drugs that would potentially correct that defect. And we found Fzero. Fzero was a drug discovered in the early 2000s. It was manufactured and synthesized in Japan for Alzheimer's disease. It passed phase 1, which is a safety trial, and it failed on phase 2, which is efficacy for Alzheimer's disease. So here we have a drug. We know it's safe because it passed phase 1. We know it doesn't work for Alzheimer's disease. And we're not surprised because when you put this type of data, big data from Alzheimer's, GRMs don't show up as significant. And we thought they had the wrong disease. We thought, they noticed mice completing, when they do their preclinical analysis in mice complete measuring better. Alzheimer's mice, whatever that means, complete maze running better. They thought it was because of memory, we thought it was because of increased attention, right? So we thought they had the wrong phenotype. And we went forward to put this into a clinical trial. We raised $1 million to basically buy the rights of this drug from Japan, 20 years old, off patent, reformulated it for ADHD, submitted it to the FDA, which is the regulatory body that controls food and drug in America, paid another set of money, and ran a 9-week dose escalation clinical trial. Lots of tests, very rigorous. And remember that 85% of patients that agreed to be recontacted? Remember, we know the kids that have these, we know the 200 or so kids that have these very rare defects that suggest they have ADHD. So we just reached out to them, asked them if they would like to be involved in this summer trial. So it's in summer, kids are washed off of their Ritalin or Adderall or whatever stimulant they're taking to control their ADHD. For 9 weeks, we do dose escalation of this Fzero. Fzero is so safe, the FDA doesn't even regulate it, right? So it's not regulated, which is in extreme contrast to Ritalin, Adderall, and all of the other speed derivatives, methamphetamine derivatives that parents put their kids on. So this is a different mechanism, a clean drug, but it works for a handful of kids. That's the problem. Anyway, this was a phase 1 clinical trial. As I said, phase 1 is for safety, but you can ask questions. And when we ask subjects, so when we ask responders, basically, kids and their parents, how they do, they respond significantly better over the 9-week period. When we ask the clinicians who are arguably less biased, they also agree that they improve significantly over the clinical trial for symptoms of ADHD. So of course, in academia, we publish this, right? Anybody has any idea how long it takes to take a drug, to develop a new drug? How much money and how much time? So the old saying used to be $1 billion in a decade. That has moved to 2-point-something billion and a little bit over a decade today. So we did this within 2 years for just over $1 million, which I think is awesome. And it's in phase 2 of clinical trials now. We'll see how it is. So that's already the take-home message. Drug discovery today is a highly imprecise science, meaning, big pharma will test 100 people having no idea how things like Benadryl work, Panadol. We have no idea how these things work, but we know they work. Antibiotics, mold on bread. We don't on and not and get something like 20% efficacy for billions of dollars in multiple decades. If you give them a cheap genetic, if you develop a genetic test using big data first, and give them a cheap genetic test, just free, you know, you have design drugs where you investigate and validate the biological pathway that's effective, and you build drugs to target that biological pathway that is effective. You can improve the efficacy, the cost, the speed, and everything. Just the overall management of the clinical trial significantly. And that's sort of what I would like to get. So, you know, we developed this, you know, there are 300,000 samples of CHOP. ADHD is the most common psychiatric pediatric disease, asthma is the most common pulmonary pediatric disease. There's a lot of autism, there's a lot of IBD, inflammatory bowel disease, ulcerative colitis. This works over and over and over again. Build it once, turn it crank, and we start spitting out drugs. That sounds awesome, right? I want to do that in Trinidad. But we can't. And we can't because of reasons that Dr. Single is about doing too. 96% of genetic studies, at least up to 2011, were done in European-derived individuals. 96%. 96% of us are not European-derived in this room. Right? So, in fact, it's well known there's huge disparities in healthcare in America. African Americans get 20% more depression, 40% more obesity, ton more diet, 30% more heart disease, and so on and so forth. It goes on and on. Right? So the entire Human Genome Project sequenced 6 or 7 European-derived individuals. Why? I don't know. Because National Geographic and many others have demonstrated by haplotype group tracing of mitochondrial lineages across the world, life evolved out of Africa. Right? Life evolved somewhere in here, and people essentially migrated out of Africa to the rest of the world. How did they get, for instance, to North America? Well, they crossed the Bering Straits. They walked to North America. It's a long walk. It's cold, it's dangerous, and the majority of them probably did not survive. So you walk across these straits, only the biggest and the baddest, and you make it across these straits. And then all of North America gets populated by a continent, right? So these are the Europeans, right? So the patterns of these Europeans, the same for Europe. So basically this entire tract out of Africa has been repeated bottlenecks in population size because of plague, because of extreme cold and temperature and virus and whatever, right? So what that means is by characterizing the genetics of essentially European-derived people, it is of little to no help to the rest of the world. It certainly doesn't help Africa. It certainly doesn't help China. It only helps European people. And similar for China, if you characterize the genetics of China, since life evolved out of Africa, there's very little overlying or common patterns of genetic variation. On the other hand, if we focus on African and more ancestral populations than Europeans and Chinese and Indians and so on and so forth, the patterns of genetic variation we find in them are likely to apply to all races because life evolved out of Africa. Does that make sense? Well, don't take my word for it. The National Institutes of Health has billions of dollars dedicated. You know, how long has it been? It's been 2003, the genome was sequenced. We're on 15, 20 years of this. And can anybody name one drug, one blockbuster drug, a paracetamol, something like Prozac, just one drug that has come out of hundreds of billions of dollars probably, and a lot of time? No? Stay tuned. Anyhow, the National Institutes of Health are spending billions of dollars going to Africa, to ancestral populations. They have, I don't know why, because there's no KFC in everyone like there is in Western Africa. There's hardly healthcare in Africa, right? So it's going to be, to me, these type of studies should be done in a place like Trinidad Tobago. Because in the Western Hemisphere, we have Western problems, and we have a lot of diversity to investigate. So you just look at this room, so Trinidad is genetic diversity. Trinidad is basically African, Indian, and mixed. There's a long tail in the distribution of other ethnicities, none of which are going to benefit from the precision medicine efforts that have been up until today. So the argument is if we don't look at our own population, if we don't collect, catalog, genotype, analyze, and publish our own data, we're never going to benefit. This gets us to the crux of the story. This gentleman is Jeff Fzero, who's a partner with me, lives in Silicon Valley now. And Jeff went on like a 23 and so do you guys understand what keeping you from the festivities outside, but when I say 23, who's sort of ancestral company around about 20 years. 23 is Silicon Valley, contemporaries around about 10 years. So we live in Silicon Valley, Jeff is 23. Right? Who can guess what Jeff's answer? Just look at this picture. Who can guess what that is? So it's 30 Who says China? Who says India? So China and India. Who says Africa? So China and India. We can look at it. China and India. Jeff has all 5 of the races. He has East Indian, Chinese, and an increasing level. He has Carib, apparently. A little bit of African and a touch of white. So in the period we sent Jeff Delta, their Jeff, as someone with African or African American ancestry, you're uniquely positioned to help advance African American. This is how Trinidad does their research, right? I don't have any people. Okay. This is terrible. And the reason Trinidad, and it's not that they don't know what Jeff's continents and ancestry is. That is easy to gauge. They don't know what to do with it. They have never met people with this kind of mix. That's what mixes. They don't know what to do with it. Because like I said, the population bottlenecks in Europe is basically the same genome with 3 changes. Here's an interesting statistic. So in terms of genetic diversity, 2 random neighbors in Africa, right, are more diverse than any 2 random people anywhere else in terms of their genetic ancestry, because Africa as a continent never really underwent these population bottlenecks, right, that basically caused the other races that caused the races to go. Before my hands, so why? So as Dr. Praxi mentioned, I identified the sense, admixture is the problem, and so that is rich in admixture, right? So anybody who just take biology will know with filial generations of these dihybrid process, right? So so let's say this is Jeff's mom, she's Indian, let's say Jeff's dad, we believe he's Chinese, I'm not too sure, but I don't know who has the card, but let's say these are Jeff's great-great-great-grandparents, right? So these 2 get together, 1 chromosome comes from the mom, 1 comes from the dad, fine, and that magic, what is it that introduces human variation into the genome? Recombination. After recombination, these things recombine, and entire stretches of chromosomes are switched. You do that a couple of times, then you get, smaller and smaller stretches of DNA that is so-called linkage disequilibrium, inherited together. And when you get to this level, maybe a small touch of this is Caucasian. The kinds of studies that have been done thus far, I'm not going to have a guy like that, or most of us in this room, because we have such a mixture. So that gave myself and a few others in this room an idea. It's like, why don't we do our own thing, our own ancestral account? Where we know there's a tribe in Trinidad and Tobago called the Caribs, there's a tribe called the Arabs. Ancestral account does not know this. Frankly, does not know this. They cannot interpret 3 and 4, we have mixture. And I got Jeff. So we are focusing as a company on this. But forget Jeff, my own story. Like, this is my parents, these are my kids. International Wheelchair Day.
It's really difficult to cut off a geneticist. I know because I'm a geneticist as well. And we get so passionate about this topic, especially when I'm going to have to tell you to table it for the panel discussion because we want to hear from Mr. Kane.
Anyhow, Mr. Kane, this has a nice story. So my dad is a red man from Tobago. And my mother was an Indian girl from Gasparilla. So I know how they met and how they went through to get together. But I have no idea. I can't tell my children what happened before. Where did you get the right from? I don't know. I don't know if it's England, I don't know if it's Spain, I don't know. And I would like to know. Right? So we will be able to answer these questions for me, and I'm sure questions you may have. And just to close, the race is on for blockbuster therapeutics. This gets serious. This is serious business. The race is on for blockbuster therapeutics. Everybody thought genomics and the last 15 to 20 years of genomics were bigger blockbusters. But there have been few. I'm going to tell you one that exists. It's called PCSK9. PCSK9 is an autosomal dominant mutation that was found in France. I have notes on this in 2003. This was published in 2003. Autosomal dominance, right? That wasn't the smoking gun. You know where the smoking gun was? It wasn't in France. It was in a rare population in Zimbabwe somewhere. They found Africans in Zimbabwe walking around with cholesterol levels. Now their cholesterol was 0. Nothing. 0. And they realized they had different mutations in the same PCSK9 gene that these European studies, that European study in France had, right? So that's the point. You can't trace the importance of this variation without diverse ancestry to compare and contrast. This race is on for a blockbuster. This is a drug for black people with high cholesterol. Every single film company is on this. Every single one has a monoclonal antibody for PCSK9. Right? And why? Because it's not a statin. It's a whole new class of cholesterol drug. And it's for African Americans. Right? So this is the type of research we would like to do in Trinidad Tobago. Because unlike this admixture, in America, there's maybe 2 or 3 generations of mixing. There's an F1 or F2. In Trinidad we don't take F6, F7, and F8 because we've been at mixing for a long time, more than most. And these patterns of variation is what Reggie and myself and other geneticists look at to map disease. 40%, a large proportion of 40-year-old Indian men drop dead from heart attack in America. Africans, they don't drop dead. So I have very simple questions. What about the others? What's going on? Anyway, before that, Dr. Carrington is in Europe. She studies dengue virus. That is typically what we as a tropical country are taught to contribute to disease, right? We have all kinds of infectious diseases that we can study here. Dr. Clarkson just told us about cancer is out of control in Trinidad and Tobago. Africans with breast cancer, none of the panels work, none of the standard panels work. So we know precision medicine is not good. But all these papers came out this year. What is most striking is that the rates of death from non-communicable diseases, cancer, heart disease, diabetes, and so on and so forth, are higher in developing countries than they are in America. This idea that we don't have data to study complex diseases is actually not true. Data does not support that. So, to close, I think...
I feel like that's the second time he said that.
I have the privilege of going to work every day, but I'm going to do in San Francisco, to do this. But for the wrong people, I want to do it right here in Trinidad. So my mentor told me to do is was on President Obama's precision medicine and action plan, right? And he likes to say data is power, data is revolution, data is frozen knowledge. And I completely endorse that statement. Most important slide. This is about 10 years of my life, plus or minus. Most recently is about Fzero UWI. We had collaboration now with Dr. Darksing, Dr. Roach. We started a company to sort of take this approach to genotype and to characterize our people, to research on our people, help map our ancestry in our localized context. And I'm Dexter Hadley. I'm the principal investigator of the Hadley Lab, and our mission is to make answers.
I want to take a moment to recognize, please come, Nigel. I just wanted to take a moment to recognize the entrance of Dr. Brian Coon, who is the Dean of the Faculty of Science and Technology. I wanted to thank you for hosting us tonight. So very speedily, I will introduce to you Mr. Nigel Keane. Mr. Nigel Kane is a businessman, right? But more than a businessman, he has a passion and a drive to see this type of research and this type of work done in Trinidad and Tobago. We want to see, and Mr. Kane wants to see that, when we in Trinidad and Tobago and in the larger Caribbean, when we diagnose a patient, when we give them a therapeutic drug, that we're giving them something that is really going to work. And because of that drive, he has started a company called Fzero Genomics. And I thought it would be appropriate to give him a couple minutes to introduce his company and tell us a little bit about what they're doing.
Good evening, everybody. My presentation is not going to be as scientific.
I don't think the mic is on. It's on your head.
Hello? Is that better? Yes. All right, you can hear me now. Sorry about that. So yes, good evening. My name is Nigel Kane, and as the panel accompanies our presentation, I want to give thanks to Dr. Roach, Dr. Haraxin, and Dr. Hanley for hosting the scientific portion of this event. And I'm just here essentially to introduce you all to Fzero Genomics, and in addition to that, let you know about the free giveaways that we'll be having at the networking session outside, and as well as introduce to you some of the services that we intend to provide the local population in Trinidad and Tobago. Okay, so Fzero Genomics, and I know that when you saw Dr. Hanley's slide, and it's a good place to say that Dexter and I have spoken essentially for over a year every day. My wife thinks that I love Dexter more than I do her. And as you can tell, Dexter likes to talk. I don't think Dexter had the conversation he hasn't. Okay, so I understand why you went over, but I'm going to keep mine a bit shorter so that we can get to the panel discussion. So, Fzero Genomics, what we are essentially is that we're going to be the first genetic ancestry testing company that focuses primarily on people in the Caribbean and the Caribbean diaspora. I think we heard from the doctors that right now, the majority of people who have their DNA sequence and genotype are of European descent. And that has resulted in a number of deficiencies and a lack of accurate data about people who have our makeup, our admixture, our look, and we are going to provide this using a digital online experience, and that is our intent. And quick history of the name of our company, Fzero Genomics. When Dr. Hanley just put up the slide with the appearance, we saw that the original gene was called the Fzero gene, and you see the connection. We're the first in the Caribbean to offer genetic services specifically targeted to the audience here, and that is the reason for the name of our company. So, as a Caribbean people, one of the things we do is that we have an affinity to our motherland. On national holidays, we celebrate, Eid, Diwali, John the Baptist, you can call them. All of them, not all of them, the majority of them are related to national holidays. Our food, as well, is very diverse. My sister, when she came to pick me up at the airport last week, she almost got told because the first thing I did when I came out of the airport was go for adults. Because I live in the States, close to Philadelphia, and I don't have adults. So you can get to and I'm the adults. We also share, obviously, cultural practices and observances, all the holidays and all festivals. And in addition to that, music. We don't only listen to one type of music, we have a variety of music that we listen to. And what is significant about that is that even though we come from diverse backgrounds and have a affinity to our motherland, we as people share together. We partake in these cultures overall, and that is significant about us as a people. And the importance of that, I'll get to a bit later on. I'll do so by a couple of stories very quickly. The first story is the story of Captain John Douglas. This is Captain John Douglas. Alright. Swaggerific, right? Back in the day, Captain John Douglas was born in 1871. And he has some style for 1871. And he was the captain of a ship that did commerce up and down the islands. So you put your produce or material on the ship and you would take it from Barbados and to Trinidad and to St. Lucia, up and down the islands. There's a couple of interesting facts about Captain John Douglas. He was travels, which at that point in time was very typical. There were no airplanes back in the day, so he, commercial-minded person that he was, captain of the ship. What I should tell you about Captain John Douglas is that he was also a very social person. And as a result of that, he was born in 1871, as I said. Captain John Douglas had 48 children. 48. You know what's interesting about that? That we know of. Okay? That we know of 48 children. I guess it's a good time to tell you guys that Captain John Douglas was also my great-granddad. I would like to know that I do not have this genetic disposition for children. Okay? I do not bear that. But that is my great-grandfather. And a few years back, what happened is that coming from such a large family, my mom was in the audience here and is one of the resident genealogists in the family. We got together as a family in Barbados, where he was originally from. And I got to meet so much of my family that I would not have met otherwise without that connection. And what was significant about that for me was that I got to understand, I have family in St. Lucia, I have family in Trinidad, I have family in Dominica, there's 48 children, so understand, I have family in the States, and I was able to make certain connections and bond familiarly with this family. And for me, it was a very satisfying and enriching experience. Why? Because I got to connect with people who are my family, but also who do not live near where I live. And that connection has remained with me, and I've kept contact with a number of members of my family, extended, very extended, and it continues to work well for me. I continue to be satisfied. And how does that play into Fzero Genomics? Well, one of the services that we're going to be offering is that currently there's a bit of an issue. Even we talk about the fact that 23andMe and Ancestry.com, who do genetic testing and probably the most popular services out there, they focus primarily on people of European descent. And what happens is that even though there are some Trinidadians, anybody here has taken, except for the Dr. Sujin, anybody here has taken an ancestry test before? Just anybody you have? All right, about 6 people, if so many. So we have 6 people who have taken an ancestry test before. And what happened is that Joel Thomas, I think, took his interest his test on Ancestry.com. And even though he took his test, Ancestry.com was not able to find much of his family. So much so that he had to go on to the internet and post a letter, said, I'm looking for my grandmother, Caroline Gomez, who was born around Captain Douglas' time. So people have to do it, right? And this is the deficiency that a lot of these services have, in that DNA testing has become commoditized. But the focus, generally speaking, is on, as you saw in the next slide, that pizza, a sliver of that pizza is represented by the majority of people in this room. And we have come here to change that. That is what I want to mention. So, using DNA testing, we'll be able to match you and your family without having a familial genealogist go out to try to track down all your siblings. And that is what our test is doing for you. And then the other story is Jeff's story. I think Jeff's story says it all. It encapsulates the fact that these companies, yes, they gave up the test, and as you can see, they got the majority of Jeff right. So it was Indian and some South Asians and Chinese. But they sent him an email and saying, hey, you're African American. Thank you, we want you to be part of the study. Imagine that. I mean, also what Jeff looks like. Jeff is a typical Trini, eats doubles, protein. He's definitely not African American. And that's a mistake that they made. That's a mistake that our company Fzero would not make, because we sound like you, we look like you, we talk like you, and we know who you are. And that's the story of us. In this room, there were 5 people, 6 people who took a genetic ancestry test. And based on our market research, both anecdotally and standard, one of the comments or feedback that came to us was, listen, I know my ancestry. I can look in the mirror and see that I'm African, but I don't really know which part of Africa I'm from. I don't really know which part of England I'm from. And I'd be very interested in knowing which part of Lebanon, or Syria, or wherever your forefathers come from. And right now, as you look at the population, we have amongst us a very diverse or admixed, a fancy genomics word I learned from Dexter. And when you look at us, the majority of us are not essentially, and I use the word pure in the sense that we have, from our forefathers, a mixture in terms of the DNA. And our company is going to allow you to come and find yourself about you. And the reason we're going to do it better than these other people is because right now their tests are not able to identify correctly people with our unique admixture. Make sense? Right. And in the future, this is going to be one of, well, the only right now. Our intent is that we are in discussions with historians, anthropologists, and genealogists to put together a genetic test and analysis that will use your DNA to tell you what both your foreparents came to this island or the other islands on. And how we do that is that you know our history is a legacy of ships coming onto the islands, whether it is during the slave trade or with indentureship, or even if it is you came over just for convenience or better opportunity. What you find happening is you'll be then able to tell you not only what your ancestry is based on a local knowledge, we're able to tell you who your family is, but eventually this test we're developing, we're going to be able to tell you what would your foreparents came over to the island on. And I think that completes the picture of who you are. To do this, we're going to be having a world-class team, and in addition to the doctors here who we work very closely with, and again, I want to give thanks to you for allowing us to work and collaborate with the team. You're going to have 2 guys that are worldwide renowned in the field of genetics. The first is Esteban Isha. And what's significant about Esteban is that he was a genetic advisor on President Obama's precision medicine initiative. Let's talk to you about that. And in addition to Esteban, we're going to have Akanasa, who right now is the director with Center Applied Genomics at the Children's Hospital of the CHOP, one of the best pediatric hospitals in the world. So our team is going to be comprised not only of people locally, but also internationally renowned geneticists. And they're going to bring you the results that 23andMe and Ancestry.com cannot be accurate. Okay? We talked about the local aspect, and this is what the initial team. These guys didn't want some of the pictures up, so I apologize to them primarily. To start with, there's Donnie. And Donnie is Donnie Natchan. So he's our Chief Technical Officer. And there is Keon Williams, and he is going to be our Chief Operating Officer. And there is Sean Anansi, who is going to be the lead and Director for Marketing and Business Development. So as you can see, our team is both international and local because we are trying to focus primarily on an audience that is Caribbean. After that, what this is going to do is outside, we're going to host a networking event, and you can join us. There's free food. I was a university student, and I know whenever free food was an offer, I was showing up. I don't care what it was. I stayed up for half an hour, but I get to eat or something, right? So there'll be some light fare and refreshments after we're done here, and what it is going to involve is that it's going to be, as I said, a networking kind of event, a conversation, come find out more about Fzero Genomics and what it is we're looking to do. You can also now reserve kits, because what it is we are trying is that we are initiating a beta test phase in our venture, in our business venture. And that starts today. And one of the things that we are going to do as well is that we are going to have a raffle. And we're going to raffle off, for the first time ever, 5 Fzero genetic tests. And in addition to that, we're also going to raffle 10 Fzero t-shirts. So 10 giveaway as well. Right? We have food and it's given it. So outside, what is going to happen is that winners are going to be announced, and the meet and greet will give you a lot more information. We're going to have a team outside that all dressed like me. We wore our corporate pose, our jeans. Get that? The jeans.
Let's see if it's easier to stop our businesses than genetics. Yeah?
I'm one more slide. I guess I'm closing. Right? Come find yourself with us. Okay? Oh, sorry. No. One more slide. This is the most important slide, actually. You're after. So, we are a high-tech company. And what we're going to do is, in order to win, get out of your smartphones and focus on the QR code. And what is going to happen is that the QR code is going to direct you to some questions. Now, in order to win anything, you must give all the relevant information. You can see that they are asterisks, red. If it is you and you back, what we found earlier on today is that you have to zoom in a bit to catch it. And if not, you can go to this URL and it will prompt you to take you right to the code. Alright?
Thank you, Nigel. You know, what I love about this symposium, and you guys can tell me if you agree, is that this is all about next generation research that is for us, by us. Isn't that amazing? I want to really thank this panel because they did an excellent job at bringing the science of genetics and genomics to us. And I want to invite them to the podium, to the stage, so that they can take a few questions. Now, I know it is 8 o'clock. And I want to be respectful of your time, and I want to say that if you have to leave at this point, we definitely understand. Okay? There are 2 mics in the rows, and we would like to open up the floor for questions at this point in time. So just make your way down to the mic if you have a question. Yes, sir. Please introduce yourself tonight. Is it on? While we're getting that started, I have to say that there has been some research being conducted throughout the Caribbean. In Bahamas, I know both Dr. Haraxing and Dr. Hadley spoke about founder genes. And in Bahamas, there has been some research that recently identified some unique genes for breast cancer. because they recognized that the majority of the persons in their population did not carry the BRCA mutation. Similarly, in a research project that was conducted right here locally at NRC, Dr. Jamil Ali and some colleagues have found some novel mutation as well in some of our old breast cancer patients. Right across the pond in Grenada, they have found unique founder genes for Huntington disease. Right? So there is a real need to conduct this kind of research in our population, especially in a population like Trinidad and Tobago, where the admixture is so great. When you look at me, you say, okay, well, she's probably, but no, probably by now at the end of this symposium, you're not going to tell me I'm 100% African, right? No, probably not. But when I did my ancestry, they told me I was 55% of African descent, I was 35% of East Asian descent, I was 10% of, I think what is it, Native American descent, and then the rest of it was Caucasian descent. I looked at myself in the mirror and I said, who am I really? Right? Yes, sir.
Good evening. My name is Lovell, Larry Lovell. I just have two questions. Anybody on the panel to answer? Something that I've been reading about a bit. One of the questions is, in the chromosome, at the end of the chromosome, there's something called the telomere. Could you just give a little explanation what telomeres are, right? And the second question is that men are made up of the X and Y chromosomes. Most times you hear about the X chromosome. Could you tell me a little bit more about the Y chromosome and why the Y chromosome has 0.24 defective, or less than the X?
So we'll let Dr. Harat Singh take the first question, and then we'll have Dr. Hadley answer the second.
Okay, so very briefly, on the ends of our chromosomes, we have these things called telomeres, which are essentially repeats of sequences. And every time our chromosomes replicate, as cells divide, have to replicate all of the genetic material in the cell in order for it to form a new cell, right? One cell divides into two. So all the genetic material has to be replicated and then split into those two new cells. Every time that happens, these telomeres shorten. They're repetitive sequences at the end of the chromosomes that are thought to have a kind of protective effect on the end of the chromosome. And every time they divide, they shorten. So you find that as we get older, our telomere lengths get shorter. And we think that this has something to do with aging, but we don't actually understand too much more.
So I could chime in. So Elizabeth Blackburn won, I believe, Nobel Prize at UCSF for investigating aging in humans. There's an enzyme called telomerase that's involved, and you can essentially gauge how long you're going to live by telomere analysis.
Is that also read? Is that the reason why women live longer, because they have more telomeres than men?
Well, not more. There is some evidence that has indicated that in women, the telomeric length might be slightly greater, but that is still under investigation.
The second question is about the Y chromosome and why it comes up short. So if I remember from medical school, you're born physiologically female until the S or Y gene on the Y chromosome flips on and makes you a man. How does a man? But you're born physiologically female. So interesting story. So what I'm sure Captain Douglas's Y chromosome is all over the curve. Right? And a similar pattern was found in the Vikings. If you look at the Iceland chromosome, the Vikings all from basically the Viking lifestyle, which was the Roman obviously men, women say that.
So the X chromosome is not found over here, the Y chromosome is found over here. You know, the reality is there's something called the pseudoautosomal region of the sex chromosomes, which the X and Y are paired almost precisely. So we're not too sure. So more or less we have all the basic physiological genes in place to function as a sex chromosome. Fragile X is an X-linked disorder, autism is an X-linked disorder. Much of the male sex-linked deficiencies, much of these sex-linked deficiencies are not known in males. Maybe it's because you don't have enough DNA, I don't know, but it's an observation. So that's as much as I know about X-linked chromosomes and genetics and inheritance.
I'll just add one more thing to that. So, yes, it's true that females have 2 X chromosomes and males have 1 X and 1 Y, but there's also a mechanism called dosage compensation, which essentially compensates for the fact that women have twice as much DNA on the X chromosome than men. So, in fact, in women, what happens is one of your X chromosomes is inactivated and the other one stays active. So it actually is not that you have, even though we have twice as much, only half of it is really active.
Did that answer your question, sir?
Yes, thank you very much. And in the meeting with one of the show, just chit-chat with people. Thank you very much.
Hi, good evening.
Yeah.
Thank you very much for the informative time. I just have 2 very simple questions. As one of the 6 people in the room who's done her DNA testing with Ancestry.com, I was able to upload the raw data to one of the other sites like MyHeritage.com and they did their other readings. So basically what I'm asking is, since I already have that done, do I have, if I wanted to be part of this, could I upload my raw data to your website or to your database rather than doing a whole new test? Which I don't mind doing, depending on the cost. So that was my next question. Since Ancestry.com orders sales all the time, from $99 US to $49 US, what is your predicted cost outside of the lovely raffle that we can partake in?
Let's let Mr. Kane answer those questions here.
Let me answer the first question. I will answer the second question. Probably you'll be able to upload your data to us and get the same local inference that you would get. But we don't know for sure. We have that on the timeline. It depends on what markers Ancestry.com may have typed, and I believe they change over time because the design changes over time. So, probably is the answer to this question.
The second question with regards to price. We are trying to have a networking session out there. You can ask us that question out there. I would have a problem answering it. But what I will be able to tell you is that we are pricing lower than a carnival costume and a premium all-inclusive fancy.
Ancestry.com, they say they don't change the roading, that they just have more and more information coming out to reconfigure. So, like when I did my first test, I was 27% of one thing, all of a sudden I'm 41%.
That has to do with
The reference that's used. So remember I told you when you were mapping variation, it all depended on what reference was, right? So essentially what's happening is more and more people having their genomes analyzed, more and more data is being thrown into the cloud in these de-identified samples, right? So we have more and more samples to which we can compare our data. And because of that, it's allowing a more precise mapping of where you may have come from. So yeah, recently, very recently, just last week or something, Ancestry.com redid analysis on all of their consumers, and people started getting quite different results than they got this time around. And this is something that is going to happen more and more as genetic tests become more prevalent, in that the data, the raw data is not going to change, but the interpretation of that data is going to change based on knowledge that we gain by participation of more and more people in these types of studies. So it's not unexpected that over time, you will have your data reanalyzed and the answer might come out more precise than previous.
All right.
Good night, everyone. So my name is Kimberly. I'm also a PhD student with Dr. Augustine. So my question is with respect to precision medicine and health disparities. Okay. So the way I see it, and this is my personal opinion, is that precision medicine is really going to benefit those people living in high-resource countries. So countries with a lot of money to spend on having this whole arsenal of drugs, because what's going to happen is that you're going to have a situation where you're going to have more drugs, and each drug is going to be so personalized that will be beneficial to a smaller subset of people, right? That's sort of the progression. So for countries like Trinidad and Tobago, most of the countries in the developing world, where, for example, for breast cancer patients, there's often times when they can't even get Herceptin, which is a WHO essential medicine, right? That's for your breast cancer drug. So my question to you is, one, why should we be focusing on this? I mean, we're a very small country. Why, with respect to our resources and our talent, money, etc., why should we be focusing on this? And how do you see it benefiting Trinidad and Tobago given our current health system challenges?
Thank you for that question.
So I'll take a shot at that. So I've had experience with that exact question. So Dr. Carrington is in the room. We wrote a paper about dengue and serotyping dengue, right? Stanford developed molecular. So dengue, there are 4 types, serotypes of dengue, and it's thought, why do they test, regions for which serotype is circulated in St. James or San Fernando or Brooklyn or wherever, or in Venezuela? Why? Because it's when the circulating serotype changes that we lose herd immunity and epidemics break out, right? The traditional test for dengue is protein-based, and it's an antigen-based test. And it develops 3 to 4 weeks after, right? You get sick and you die from dengue within 2 to 3 weeks, let's say. And weeks after, you're going to get serotype results. That's how it's been done, because of the surveillance, because of the epidemiological surveillance. So we can turn that into epidemiological surveillance of 1 with a molecular test. You can just remember, my kid had dengue 1 this year, my kid had dengue 1 this year, my kid had, he has dengue 2 this year. Let's pay more attention, right? So even in resource poor, and I wouldn't consider Trinidad resource poor, but even in developing countries, the precision takes epidemiological surveillance down to you, and you cannot. And dengue is just an example. The other side of this is that medicine is generating more and more data. Genomics is just, I read all kinds of data. Genomics is maybe 5 years away from genomics, but there's more imaging, there's more markers, there's more drugs, more and more and more, right? It is becoming humanly impossible to deal with the deluge of data. It's not possible to go learn all of medicine. You have to learn how to discover medicine, how to search medicine. And the reality is, and there's like Trinidad Tobago, where there are cases of brain injury, where people leave here and they don't come back with knowledge that they've learned in med school or engineering school or whatever. At least in medicine, if we develop this technology, albeit in developed countries, albeit properly sample the populations so that we learn not biased results, but real results, you can imagine that can be exported at scale to Brazil, Russia, India, China, and Russia.
Can I add to this? I think that is a very valid question, and it's one that I have also probed myself, and I think you know that. Because in Trinidad and Tobago, where we have such a huge burden of chronic disease, and our health system, many would argue, has been failing us for some time, one might ask the question, why make such an investment in something that Dr. Hadley himself has told us will take years to see some sort of benefits? Right? That's the essence of your question.
Yes,
this is my personal opinion. I cannot substantiate it with facts, but I can tell you that the trajectory of other developed nations, right? I think it is important in Trinidad and Tobago that we realize that both of these things can and should be done simultaneously. We should invest money in fixing the root cause of the issues in our public health system. But at the same time, we cannot be left behind this precision medicine and genomic era. If we are left behind, as we have seen, that my colleagues tonight have spent some time to explain to you, if we are left behind in this era, the drugs that have been created would have been created for us, and it will not work for us. So, what we need to do is we need to take some time and really have a true discussion and a true analysis of this very issue and say, look, what are the issues we have in the health system? What do we need to invest money to fix immediately? And how can we look ahead at the same time? We can't do one without the other. It's just not going to happen. We will be doing ourselves a great disservice if we do that. Does that answer your question? Lovely. So I think we have the next question. He's been waiting for a minute.
Good night, everyone. My name is Mrs. Anna /Hunt. And this whole thing is very exciting for me. I used to follow Ancestry.com. What I want to find out, I have two questions. One, what I want to find out is if your company, but I don't know if it has already been formed as such, I got the idea that it is a working progress, that is formed.
I think it's a work in progress that has some concretized elements.
So, in other words, we are bringing Ancestry.com closer to us and at a higher level.
We're bringing the best tests for people in the community.
Right.
To us.
I'm thinking now of families and persons who may have had a challenge or challenges in health where, and I take one specific, in looking for a match, a compatible match, whether it's a transplant, blood, and so on. Would you be able, with your company, to facilitate that with your skills and so on at a faster rate and at a reasonable cost? That's one question, if you're going in that direction.
So I do that now, essentially. So we have large medical. So I think with time, yes. I don't think Trinidad and Tobago's public hospitals will be on paper forever. They're going to move to electronic medical records. They're going to have large numbers of electronic medical records sitting there. And if there is a genomic platform to at least capture the molecular data to tie it to set electronic medical records, you can begin to do the kinds of things that you're asking. And this is exactly what we mean. There's the idea of predicting a donor predictor score, meaning we can look at all the labs and all the drugs and demographics and the genetics and everything to do with people that have died and are going to give up their kidney and predict, is this going to work or not? If it doesn't work, find somebody else. Right? So these are the kind of things that are happening today, and inevitably they will happen.
Okay, and the time frame, can you give us an idea of the time frame?
I personally cannot give you a time frame, but you can come to San Francisco and I'll show you what it's like.
All right, lovely. So I think we
last question very quickly. I have been reading, and I'm assuming it's not fake, that you will see two persons who look alike and they are not related. How is that explained from a genetic point of view? All right, I'm sure you have read and seen pics. Two persons, different countries, different experiences, different families, and they look even more like that identical twins. How do you explain that?
10% of kids don't know the biological clock. It's one way to explain it.
So you concentrate in Jamaica.
So you're saying that when that happens and comes up, what you mean is not true.
That's nice.
They are twins.
But I think the explanation.
But you understand what I'm saying?
Yes,
Where that come across and so on.
The answer in genetics is not a linear process. There's multiple pleiotropy, there's penetrance, there's all these features on DNA to RNA to protein that qualify that process. And there's just luck, dumb luck, right? So if you have 3 billion base pairs, hundreds of millions of splice forms of proteins probably in a full human complement, there is the chance, again, I'm not a mathematician, but there is a chance you can just look very similar to somebody just for chance. So there's a lot of noise in the genetic system, and many ways you can get to the same conversion, whether it's drug, disease, or looks in this case, height, and whatever. There are many ways and many effective routes one can take to get to the.
So I'm going to ask that the questions are phrased very quickly, and that the panel does us a favor and answer these questions very concisely. Yes?
My name is Dr. Nicole Ramlichan. I am a geneticist at UTT and at Dr. Ali literally a few years ago. And I have my other panel members. So we say welcome to Fzero Genomics. I'm a consultant at Genetic Diagnostics Limited, who's doing a little bit of what the lady before was asking about in terms of health genetics. We do a lot of screening globally for cancer, cardiovascular, diabetes, those kinds of things. There's a lot of data coming out that's really exciting because of the diaspora and the diaspora nature of the country. In addition to ancestry testing, to kind of get around, we offer ancestry testing and have been doing so for about 5 years, but we run into a lot of trouble that you know about because of the tests that are available. So we actually have had to build our own database from scratch and things like that. And it'd be really exciting to have you all here. Very excited to hear about it and excited to meet with you all.
Thank you very much.
Thank you.
The geneticists are coming out of the woodworks. Yes.
So, as a computer scientist, I'm super excited about the data that comes out of all of this and what we can do with it. But as an individual, I was just hoping someone could touch on how is this data going to be used? Should I be nervous about maybe not getting a job because of the data that I'm giving for some of these studies? How is that being taken care of, either commercially or through the university studies or any of these aspects?
So yeah, so they've just thrown me in on the spot. But I think your question is a very good one. One should be concerned about where your data is going and how it would be regulated. What I can say is that in many of the more developed nations, there are specific laws that regulate how data is used. While in the Caribbean, many of our islands do not have similar laws in place currently, there is a drive towards creating those types of policies to regulate the data. Nonetheless, at present, there are mechanisms in place that can protect the individual. And through a university, for instance, if you participate in a university study, there's an ethics committee that would review that study and does have a discussion with the researcher about how the samples and data can be used. But believe it or not, the person who has the greatest say about how your samples can and cannot be used is the individual. And that is sort of encapsulated in something that individuals sign when they participate or they donate their samples, and that's something called a consent form. Nobody can use your data in a way that you have not specified or they have not asked you in your consent form. I think Dr. Hadley had shown a slide where 85% of the individuals had agreed to be recontacted. That's an example of someone exercising their right. You can say, hey, if you're going to use my sample, I want to be recontacted so that I can give you some indication. Or some people are very comfortable and they trust the researchers and they trust the institutions to which the researchers belong to protect their sample and protect their privacy and confidentiality.
Alright, so let me just add a bit of color to that, because obviously that question is relevant to what it is we're doing as well. And it's a good place to let the audience know that we, as a company, will be implementing the same developed countries' laws and practices with regards to your data. And I think it's the point that Dr. Roach made about who owns your data. You actually own your data. We as a company are not going to do anything with your data that you do not allow us to do. And I think that that's a significant takeaway. That informed consent process is very important with regards to our stewardship of what is your data. And you control it, and we only do what you want us to do.
So we take 2 more questions, and then unfortunately we have to break out. So, yes?
Good night. I just have a few questions. Just straight.
2 more questions.
The first one is.
Speak a little louder, please.
First one is, how expensive will be the cost of the testing?
But as I said before, just to remind everybody, cheaper than the price of a Canada costume and a premium all inclusive. Be sure.
How long does it take to get back the results?
So right now we are launching, as I said earlier on, our beta test phase. And the beta test phase allows us to basically test all our processes to ensure that they are accurate and functioning efficiently. So what you'll find is that, generally speaking, for the beta test phase, our tests will take about 8 to 12 weeks. The results will come back to you. But when it is launched, we're looking at somewhere between 6 to 8 weeks to get the results back to you. You, however, will be notified as the process goes along as to where your sample is in the process.
And the last question is,
one question to 3 parts.
last question is, in terms of, you mentioned the biobanks. So my question is basically, what kind of jobs are offered in the biobanks, and at your company, what jobs?
So I think I agree with you. I'm hearing some agreement or some consensus in the room that we need to have our own biobanks, right? So I think if we allow this company some time to grow and to really get a sense of the needs of the population, they will design themselves in a way that is reflective of our needs. We can agree to that, right? So who knows, maybe a biobank might be their future. But I don't think it's this company is at a place at this moment where they can answer that question definitively.
I beg to differ. And I do it very respectfully because Alana is our scientist, and we try not to talk business with our scientific advisory team. But our intent is, and that is through conflict of interest, not because we don't want to talk to them, but through conflict of interest. That we have to do.
There are some things I just cannot know.
Amanda is our ethicist, and some things we don't tell them because like, no Nigel, no Dexter, you guys can't do that. But that being said, our plan is definitely to have a ban. There's no reason to get into this. If it is, we are not going to explore genomics and genetics to its fullest. There is no one right now that is concentrating on us as a population, because there are 350 million people alone in the States.
You see, I was trying to be diplomatic because Dr. Harakson and her lab have been doing a great job thus far, I think, of collecting samples and trying to create a small biobank.
So as a researcher, we are also trying to create a biobank so that we can have various studies ongoing, and some of my students are up there in the back. They're the ones that are actually going out, Akili, Alana, Royan, they actually go out, Kimberly over here, they actually go out into the various health centers and the various RHAs, and they collect samples They do informed consent on the patient. They sit down, we have a conversation with them. They ask them, are you consenting for us to do X, Y, and Z with your sample? We store their sample, and then according to what they say, we use it, in that.
So I think the big question will be, how can we all work together to create one biobank that is going to serve the country? Yes? I think we can agree to that.
I'm just chiming in. That is late for this country. This has been going on for a long time. Iceland, in Estonia, in England, in China. China has the biggest nationalized biobank in the world. And Chinese genetically cannot be China. Somebody thought about that, implemented those regulations. And that's how it was.
In Guyana, actually, their samples, they have similar stipulations on their samples. They have a biobank in Guyana, and their samples also cannot leave the shore. So there are things happening in the Caribbean. Trinidad just needs to get on board.
So the point is that many examples to look for to tailor a plan, regulatory plan, a science plan, privacy plan, security plan, and so on and so forth, to take care of it.
Thank you. Yes. Final question for the night.
Yeah, just have a quick question related to the rare illnesses. I think it was touched on Dr. Harris' what are some of the rare illnesses that we should be looking at in terms of testing? You mentioned common illnesses and common variants, delivery variants to physiology, and will our population decide how sufficient in terms of testing for rare illnesses? And what I know is that part of the Fzero Genomics pitch also in terms of rare illnesses and chemical illnesses and these things. Thank you.
Okay. So to answer your question, a rare disease is a disease that affects, I think it's 1 in 100,000 people. The frequency, something like that. And while that seems quite rare, and our population is so small, we do in fact have a handful of people who do have certain rare diseases. Rare disease is a term that encompasses a large number of different unique conditions. Many of them are neurological or neuromuscular in nature, but they tend to be caused by a single mutation that has a really strong effect. Rare diseases are actually one of the easier types of diseases to study genetically because of that fact, because they're usually caused by a mutation that has a really strong effect. And so a lot of times it's simply a matter of looking at the parents of that child and the affected child and mapping variants in their genome and trying to figure out what's the cause of that disease. Now I've been doing this for a long time with the Fzero Genomics Institute. This is a non-profit organization, and we do this for rare disease patients around the world. And that's actually one of the, I would say, triumphs of this whole genomic era, that we are actually able, in the case of rare diseases, to be able to resolve what these are. Now, in the case of complex disease, like the ones we were talking about, they're much more, when we say complex, not only complex in terms of their physiology, but they're also complex genetically. And so that makes it a lot harder to study them on a genetic basis.
Okay, at this point I'd like to, ask the panel if they have some closing words.
I just wanted to give them a little bit of insight. Health genetically is not something that we are focusing on in the short term. We have to explore what Trinidad looks like, but it's definitely an initiative strategically that we are looking into to assess its viability in this population. But as I said, if it is, we are here to do genomics. We intend to go all the way.
Yes, so I'd just like to ask the panel if they have some closing words. Nigel, you want to start?
Yes, I'd just like to thank everybody for attending. I'll be honest, when Dex and I and the team first talked about putting on this symposium, we're like, how many people are going to come to get to listen to a conversation, a discussion on genetics and ancestry mapping? And this number far exceeded our expectations. I know you took a lot of time out of your day to attend, and we really appreciate that, so much so that after the event, you can come and look with us and have some light there and refreshments as well. So thank you very much for attending.
Thank you. Virginie, any last?
My last remarks are just simply, these things are going to start coming at you, and we don't want you to be fearful of participating in these kinds of studies, and we want you to be excited, but also cautiously optimistic, right? So I think, and again, I would like to echo what he said. I'm really pleased to see that so many people turned out here tonight to have this conversation, and we hope to have more of these types of conversations with you, because this sort of thing is inevitable. It's happening, and we need our population to be informed and educated about how to make these decisions that have a lot to do with you and your health and your information, because your genome is ultimately your own data.
That's it.
I'd like to mirror the other panelists. Thank you all for coming. This has been a great turnout by our expectations. And I'd just like to say Trinidad and Tobago is a tiny country. 1.3 million people. So that's 25. I don't know how that is possible. China is almost double this population, and we are the same, but suffice it to say it's some number of people around the order of a million. We've given you the only instrument in the last century, apparently, the steel pan. Trinidad has the highest number of artists per capita, the greatest show on earth. We've given the world so much from this 1.3 allegedly million people. I hope what you will take away from this panel is that because of our history, sometimes brutal slavery, indentured laborers, and if you read from Columbus's journals, they're the most brutal material you can read, and this is who we are, right? And because of that, because of our diverse ethnic origins and ancestry, we have a lot more to give the world in terms of genetics. Like I said, in California, everybody seems to be mixed, but they mix one and two generations. Trinidad is mixed since 1845, and then go Pater's Act, and we all didn't keep up. Right? People like me, about 20% of our population, so-called doublets. What are these doublets getting? Are they getting this disease that Africans get, or that disease that Indians get? I think that is the point that has become quite obvious to me. And I'm the most cynical person when it comes to precision medicine. We can talk later, but I think a country like Trinidad Tobago is in a position to give you a lot more in that context.
On that note, I'd like to thank our panel, Mr. Keane, Dr. Harat Singh, and Dr. Hadley for such a vibrant discussion. I also want to thank you guys for being such a gracious audience and indulging us way past our due. At this point, we want to invite you guys outside for some light refreshments. Thank you so much. Don't forget.